In our last lab, we investigated the effects of substrate concentration, hydrogen peroxide, on the rate of the reaction with the enzyme catalase, which was found inside of a eukaryotic cell, yeast. This week we'll be using the same model organism, yeast. It's a eukaryotic cell, much like our own. And we will also be using hydrogen peroxide, but we are going to keep peroxide at a constant concentration this week. Instead, we are going to vary temperature and see how temperature affects the rate of the reaction. This will show us a lot of very important fundamental concepts of both chemistry and biology. It'll show us what enzymes do. It'll give us an insight into the importance of homeostasis. And possibly we'll see what happens when an enzyme is denatured. So what you're going to do is you're going to cool off or heat up both yeast and peroxide in separate test tubes. So remember, yeast is an organism that contains the enzyme catalase, and you are going to cool off a solution of yeast or warm it up. You're also going to cool off or warm up a solution of hydrogen peroxide. And once they've reached the temperature that you want, you're going to let them incubate for about 5 minutes. Then, while they're still warm or cold, depending on your treatment, you're going to mix them together and record the rate of the reaction. So let's say we're testing 0 degrees Celsius. You would mix ice water with a little salt probably in a beaker and place a test tube of yeast into that concentration, or into that ice water bath. You would place a test tube of peroxide into that ice water bath and put a thermometer in them. Once they've reached the temperature that you want, you'd start a stopwatch for 5 minutes. After that had counted down to 0, you would mix the peroxide in with the yeast and record the change in pressure. So our hypothesis will look something like this. You'll have catalase activity and temperature. And your temperatures can range this week. This might come out a little funny. I'm writing with a mouse, but we have a range of 0 all the way up to the possibility of up to 60 degrees Celsius. So you'll want to think about, how will temperature affect the rate of the reaction? Remember, molecules are in motion, and different temperatures will affect those molecules in different ways. And some temperatures may even affect the shape of the enzyme. We may denature that enzyme just like we denatured the enzymes or the proteins in the membranes of beets in our first few experiments. So you may get no change, which would be a straight line. Maybe you think the reaction will start off fast and slow down. Maybe you think there will be a linear relationship in this direction. It's up to you. Whatever you think will happen, you will write your hypothesis and you will test it and investigate the relationship between catalase activity and the temperature of the reaction.